Friends, welcome to Biology Made Easy. Today in this video, we are going to discuss about linkage and recombination. So first thing we must know that when Mendel conducted his experiments in garden pea, he took 7 pairs of characters. And luckily, the pea plant has also 7 pairs of chromosomes. So most of the characters are present in different chromosomes, or even if they were present in the same chromosome, they were far apart from each other. That means they asserted independently. There was no question of linkage. But when post-Mendelian scientists they started working on other characters in other organisms, they found that the Mendelian dihaprid ratio 9:3:3:1 ratio is deviated. Other ratios are coming. So what is the reason behind it? When scientists tried to find out the reason behind it, they found that the genes which are located in the same chromosome and very close to each other, they are linked together. While the genes which are far apart from each other, they can undergo recombination or crossing over, and they can be separated from each other. That means the genes which are present in the same chromosome and close to each other, they are linked. This is known as linkage. Now let us explain it in the form of a diagram. Suppose this is one chromosome, and here capital A is there, and here suppose the capital B and capital B is there. This is one chromosome. And in the homologous chromosome, suppose only recessive genes are there, small a, small a, small b, small b. Now, what may happen? There is every possibility that there will be recombination. Now you see how exchange is taking place. Here it is capital A, and here small a, small a. Here capital B, now capital B is this side, small b is this side, and this side small b. So in the new one, what will happen? Finally, it will look like this. Now this portion will be exchanged. This portion is exchanged. That means this will be capital A, but this will be capital B, while this side it will be small b. Similarly, in the other chromosome, this part will be red, while this part will be blue, and the other one is like this. That means it is small a, and here small b, but this will be capital B. That means you see between the two homologous chromosomes how the crossing over has taken place. Why it took place? Because they are far apart from each other. But suppose in one case, suppose this is a chromosome, and here gene A is there, and here gene B is there. They are very close to each other. So the probability of this recombination is very less. So in that case, we can tell that the A, B, they are linked together. So now it is clear to you what is linkage. That means when several genes are present in one chromosome, they tend to inherit together. This is known as linkage. Now the linkage was first studied by Bateson and Punnett in case of sweet pea, that is Lathyrus sativus. But later on, the details were worked out by T.H. Morgan in the fruit fly, that is Drosophila melanogaster. And for this work, he was awarded Nobel Prize in 1933, and that is why T.H. Morgan is known as father of experimental genetics. And because he worked it in the fruit fly Drosophila, he is also known as the flying man of genetics. So T.H. Morgan got Nobel Prize in 1933. He is known as father of father of experimental genetics, and he is also known as the flying man of genetics, because he worked on that fruit fly. So what is his experiment which gives some idea regarding linkage? So let us discuss what is the experiment. Drosophila was taken for the experiment purpose by T.H. Morgan, and he selected some characters. The characters are, this is body color, then the dominant character, dominant, or we can call it as wild type, and the recessive character. If this is character, and our character, the first one is body color. So the body color, it may be brown body or it may be yellow body. Now yellow body is designated as Y, while the dominant will be designated as Y plus. Similarly, if we take the eye color, it may be red eye or it may be white eye. So white eye is written as W, and W plus is red eye. Similarly, to the third character, that wings, and wings may be normal or may be miniature. So miniature is written as M, so normal is M plus. So these are the three characters chosen by him. Now taking these things, how he made the different crosses, let us see. In one of the crosses, suppose let us take cross A. What is happening we'll see, then we'll take another cross. In cross A, he took one, this is yellow with white, and another with brown and red. So this is Y plus, W plus, and the other one is the male one. This is the male one. So now see, this is the female side, this is the male side. So if these are the parents, what will happen in the F1 hybrid? You see, the female will produce only one type of gamete, while the male will produce two types of gametes. So in the F1 generation, what are the possibilities? Now this is Y, W, then this will come to this side, then Y plus, W plus. And what is the other side? Now this side also, it is Y, W, and the other is that male one, this one. So what is happening, you see? Now this is yellow and white. This is yellow and white, while this is brown and red. That means a brown body and a red eye. But here what happens? Here it is a brown body and red eye. Brown body and red eye, because both dominant characters are there. While this one will be yellow body and white eye. So this is the F1 generation. In the F2 generation, what happened? When these were intercrossed, in the F2 generation, the percentage of parental types and recombinants were completely different. In the F2 generation, from this cross, the 98.7% were parental types, while 1.3% were only recombinants. Now what is the meaning? That means in F2 generation the parental types means yellow, white and brown, red. These two occurred in the 98%, 7%, while the recombinants means a brown body with a white eye and the yellow body with a red eye. They occurred only in 1.3%. Now let us see what happened in the second cross. Now in the second, let us take cross B. So in cross B, that is it has taken these two characters. So there is it is taken the WM means white eye miniature. WM, white eye and miniature wings. Then the other one is W plus M plus. W plus M plus means red eye and normal, and the other one is the male one. So now you see, this is the female side, this is the male side, and this is white eye and miniature wings. While this one is red eye and normal wings. Now just like this, what will happen? Now this female will produce only one type of gamete, that will be W and M, while these will produce two types of gametes. So in the F2 generation, what is happening, you see. In the F2 generation, one will be WM and the other one will be W plus M plus. While in this case, one will be WM and the other one will be the male one. So in this case, what is happening, you see, this is the F1 generation. So in this F1 generation, this is W plus M plus, these are dominant characters. So it will have red eye and normal wings. Red eye and normal wings. While here W and M are there, so it will be white eye and miniature wings. Now in the F2 generation, what happened? In the F2 generation, the parental types were 62.8%. These are the parental types. While the recombinants were 37.2%. These are the recombinants. That means the parental types. Here the parental types are white eye with miniature and red eye with normal wings. They occurred in 62.8%, while the white eye with normal and red eye with miniature, they occurred in 37.2%. So if we compare these two, what will be the conclusion? What is the observation from this? That means here the parental characters are more. That means yellow body and white eye, they are very close to each other. They are linked, and the recombination is only 1.3%. While in the second one, the white eye and miniature, they are slightly far apart, so that 37.2% recombinants are occurring. This is the major work done by T.H. Morgan. So from this, what are the different observations? From this work, Morgan and Castle, they put forth one theory. Morgan and Castle, they put forth the chromosomal theory of linkage, given by Morgan and Castle. So what are the important points? Now, number one important point is that genes are present in chromosomes which behave as a single unit. Genes which are present in the chromosomes, they behave as a single unit. The second point, how they look like? They look like as if they are beads on a string. Beads on a string, like this. They look like beads on a string. Number three, the genes which are far apart from each other, they undergo crossing over. Genes far apart undergo crossing over. Then the next point, where this crossing over takes place? Crossing over takes place at certain points known as chiasma, plural, singular is chiasma. So crossing over occurs at chiasma. Then the next point is, what is the probability of a crossing over? That depends upon the distance between the two genes. That means the strength of the linkage. Strength of linkage is inversely proportional to the distance between two genes. Inversely proportional. That means if the genes are far apart from each other, the linkage is less. If they are very close to each other, then the strength of linkage is more. And from this experiment, what is another conclusion? So from this, the another observation is the next point. That means the parental characters in this experiment. The parental characters, yellow body and white eye. Parental characters, yellow body and white eye are tightly linked, and recombination is only 1.3%. Whereas in this case, that is the eye and the wings. That means the white eye and miniature wings, they are slightly far apart from each other. So the another conclusion is, that means the crossing over, it is a minimum. Suppose there is no crossing over, then it is 0%. And the crossing over will be maximum 50%. While in case of linkage, that is a maximum linkage will be 100%. If there is no crossing over, then linkage is naturally 100%. And the minimum linkage will be 50%. That means the linkage varies from 50% to 100%, while crossing over varies from 0 to 50%. This is the rule of linkage and recombination. So this is the experiment conducted by T.H. Morgan and the observations, the conclusion from it. Then the next point that we'll discuss is linkage groups. Now already we have discussed that the genes which are present in the same chromosome, they are inherited together. That means that forms a linkage group. So how many linkage groups will be there in one organism? It depends upon the haploid set of chromosomes. Let us take example. Suppose in case of mucor, one fungus, the haploid set of chromosome is only 2. That means the linkage groups will be only 2. Take the case of Drosophila. Here there are the chromosome number is, the haploid set is 4. So naturally the linkage group will be 4. That means if we take the organism linkage group and haploid set of chromosomes. So let us take the example of mucor, a fungus, and it has 2 linkage groups. Why? N is equal to 2. Then the Drosophila, which was studied by Morgan, here there are 4 linkage groups. Why? N is equal to 4. In case of a pea plant, Pisum sativum, there are 7 linkage groups because N is equal to 7. In case of a maize, there are 10 linkage groups because N is equal to 10. And in case of human being, there are 23 linkage groups because N is equal to 23. That means always the number of linkage groups correspond to the haploid number of chromosomes. That is the linkage group. Then the other things that we will discuss is the factors which affect linkage. Factors. One factor is age. With the increase in age, usually linkage decreases. Similarly, temperature. With the increase in temperature, the linkage decreases. And the third one is X-ray. If in this case also the linkage decreases. In case of S, there is a decrease in the linkage. Linkage decreases. The linkage increases. That means the crossing over is more. But in other 2 cases, there is a decrease. In these cases, there is a decrease. Decrease. So this is about the different factors. Then another important thing. What is the significance of this linkage? Significance. Number one significance is the linkage. Linkage is a deviation to independent assortment. This is number one. Number two, since in a linkage, the genes are which are present very close to each other, they are inherited together. That means the characters can be maintained. Characters are maintained. Suppose 2 good characters are there together, so naturally that will be together. But suppose 2 to bad one good character is linked with one bad character, then that is a headache to the breeders. That is the next point is a headache to breeders. How? Suppose in a mango plant, the size of the mango is very big, but it is sour because both are closely linked. But the breeder will always try that the size of the mango should be accompanied with the sweetness. But if they are closely linked, then they cannot be separated. That means habitation cannot be made. This is the main headache, not only with the plant breeders, with also animal breeders. That is one problem. So this is the significance, and another also is there. The location of genes can be determined. Location of genes. What is the exact position in which chromosome they are present, that can also be located. Then let us close the video with the MCQs. So let us discuss the MCQs here. Now one MCQ is the lack of independent assortment. Lack of independent assortment in a Drosophila. In a Drosophila is due to one choice repulsion, second choice crossing over, third choice linkage, and the fourth choice you can go for the recombination. This is one question. Let us discuss the second question. The total number of linkage groups. This is a very good question. Groups in Escherichia coli. In Escherichia coli is one choice 1, second choice 2, then the third choice 4, and the fourth choice 7. You will be able to answer these questions if you will think a little bit. So thank you very much for observing the entire discussion. Thank you very much. God bless you. Have a good day.